Level 3 sequencing. I am wondering, can it be determined in which order the nucleotides occur in every type of organism? Is there a way for humans to identify from a piece of material from a specific organism the sequence of its bases? Although I could read the sequence, an automated system would be clever, because reading the whole DNA of e.g. a human, which has 6.4 billion letters, would take me too much time. There exists a technique for determining the DNA sequence of an organism. This technique is called DNA sequencing. Using sequencing, one can determine if a DNA stretch originates from a house, human or gorilla just by reading out the sequences of the bases. DNA sequencing is the process of determining the nucleic acid sequence, the order of these different nucleotides in DNA. It includes any method or technology that is used to determine the order of the 4 bases: adenine, guanine, cytosine, and thymine. The first person who determined the sequence was Frederick Sanger. In the 50s, he determined the sequence of amino acids in a protein. He showed the order of the 51 amino acids, the small building blocks of proteins of the insulin protein, which was never done before. A bit later, in the early 70s, Sanger developed a method to determine the exact sequence of nucleotides within a gene. This method is called the chain termination method. In the same year, another chemical method of DNA sequencing was explained by Alan Maxam and Walter Gilbert. The chain termination method is still used in laboratories today for smaller stretches of sequences. However, nowadays there are cheaper and less time-consuming methods. I am impressed by the timeline of the development of sequencing. The Sanger method is tedious and time-consuming. Other methods were developed afterwards. For example, Illumina sequencing, which is part of the so-called second-generation method. Around 2014, nanopore-based DNA sequencing was developed, which is part of the so-called third-generation sequencing. Nanopore sequencing is one of the latest methods of DNA sequencing. It was developed by Oxford Nanopore and represents a new generation of high-throughput sequencing technology. It measures the change in voltage when the DNA is transported across a membrane through a tiny pore. In this picture there are few components: membrane depicted in gray, nanopore in blue, motor protein in purple, and the DNA strand in light blue. A membrane has a simple structure in which proteins can be incorporated. The incorporated protein is called nanopore. There is a small hole inside this nanopore the size of a nanometer, hence the name nanopore, such that a DNA strand can pass through this hole. The basic idea of nanopore sequencing is that in the channel of nanopore, therefore, how much exactly the DNA blocks this channel depends on the nucleotide sequence. Here you can see the scheme of nanopore sequencing. I decide to participate in this process in order to learn more about how it works. As I am a tiny guy, it is not a problem. I hook on to DNA. I jump onto a motor protein at the time a DNA binds to this motor protein. The motor protein is used to move DNA forward. In other words, it is an engine which reduces the speed of DNA passing through nanopore. It is important to slow down the DNA strand because, otherwise, the electrical signal would be distorted by combining signals of several nucleotides. Sitting on the motor protein, I start my wonderful journey while the motor protein looks for a nanopore. The motor protein is fixed to the nanopore and then opens the double-strand DNA, directing one strand through the nanopore, in which bases are read one by one. I jump to the DNA strand because I am curious what is happening with this moving strand. When it passes through, the tension on this pore is changed. The change in this voltage is specific for each of the 4 nucleobases, whereby the sequence can be recognized. When I am going through the pore, I will disturb the signal of the bases. A special feature of the offered nanopore devices is their size. Most of these devices are the size of a Mars bar and can also be connected directly to a computer. In this picture, there is a portable real-time device for sequencing called MinION connected to a computer for direct transfer of sequencing data for further analysis. Sample preparation can be simplified so that sequencing can also take place outside of a laboratory. Thank you for watching.